Hello.
Hi. I'm just going to try and share my screen.
No worries.
Oh,
here it comes. Yep, got it.
can you see?
I can see what's organic chemistry, chemicals on Earth, the first slide.
I'll just flick through. Actually, what I'll do, if I do...
Perfect. Sorry, there's a little dog beside me. Yeah, moving. Moving.
I just want to quickly... I just want to quickly go to this one and just see if this works.
No problem. I've got sound.
You can hear that?
Yeah, can hear and see it.
Okay. And then the other one is this one.
Yeah, sound.
Cool. Okay.
Perfect. Right. So we've got Claire Watkins in with us. She heads up science at college. So when we start, she'll do a brief introduction and then she'll hand over to you.
Okie dokie. What time is it now, by the way? Oh, it's 10 past, is right?
Yeah, we've got 5 minutes. Just before quarter past, I will let the attendees in, just so they can sort of start joining. And then Claire, I'll give you a shout when we've got sort of more than 20 people or so.
Okay, that's perfect. Just so you're aware, I'm in a classroom, so I will.
Oh no, probably... you're fine. We're quite used to that.
I'll pop myself onto mute
as well.
No, that's fine. My guys are going to be watching along.
Amazing.
So you'll have another 15 or so to add to the numbers that have seen you. Are you expecting any questions throughout the presentation, or is it just introduce and then presentation?
I was just going to go. I've probably got too much, to be honest, but I thought probably better have too much than have too little.
That's fine. Just so that I don't want to sort of interrupt you if I don't need to in that thing. So I'll literally just introduce you and I'll let you crack on with it.
Yeah, sure.
If there are any questions, they'll pop into that Q&A box underneath. You'll see sort of a notification pop up.
Yeah.
Okay. The only thing I was going to ask is, it's probably too late now anyway, but is if you could have done a kind of almost like a vote, if people could kind of, they could vote.
We can ask a question in a poll for you. We can sort that out quite quickly.
So that I was going to ask them with the Blackadder, I was going to ask them if they, because I asked them is Percy a alchemist or a green chemist? It'd be interesting to see which way that goes. If they think he's an alchemist. Okay, there's no right or wrong answer.
Can really. It says enter a title for this poll. So on what would you like me to pop in?
So if I go, can you see the slides again?
Yeah.
Can you see this one here? It says, is Lord Percy an alchemist or a green chemist?
It says what flavours are chemists? What flavours of chemists?
Yeah, and then on the right hand side in the kind of middle of the.
Okay, is Lord. So just copy that.
Yeah. I mean, if it doesn't work, it's not the end. I'd just be interested myself to see which way they think which way it goes, because there's no right or wrong answer, but it's just a kind of just a little game, I guess, really.
Oh, help if I spell.
I guess you can't see my cursor. Is that right?
Give it a wiggle. No, I don't think so.
I'm just seeing if I can get it. No, it doesn't seem to want to. Because it would have been useful for me to be able to point out things on the screen, but
you can get like almost like a lasery cursor mode. I could see it obviously when it wasn't in the presenting version.
yeah, I'm on Keynote, so I don't know if it's got a.
If you
So what
got
will
a
they-
top across the top, it's a little green bar, and then it says View Options. I don't know if you'll be able to fiddle in there.
So in the which one? Sorry. Oh, in the green bar, yeah. Okay.
Yeah, are there any options in there?
Remote control, more. Share computer sound. Hide floating, hide final meeting, record chat. No, it doesn't look like. I'm worried that if I change something, it all goes wrong. So I'm probably just going to leave it and just go with it as is. So do they see just the screen, or do they see me as well?
If you pop your video on, they would see you.
Okay.
And if not, they just see the screen.
Maybe it's just easier.
So I don't know if you can see, if you've got the polling option at the bottom, that's in now. If you click polls, and then if they want to use the Q&A to ask a question, they can. So Claire, it might be worth, when you do your introduction, just say a bit later in the quiz, you may be invited to complete a poll.
It's really, I mean, it's really just a, you know, it's no big deal, really. It's just, you know.
Yeah, it's there. So if you.
Oh, I can see it. Yeah, I've got it now. Yeah, see it.
Perfect. So I'll close that off at the end. That's there if anyone opens it when instructed. So I'm going to start letting everyone in now. So Claire, I'll just give you a nod when we're ready for you to start introducing. Is that okay?
That's absolutely fine. Can I just double check that I'm pronouncing your name correctly? It's Dr. Sean Bew, is that right?
Yeah.
Yeah.
Yeah, that's right. Yeah.
Brilliant, thank you.
Thanks, guys. Right, I'm going to start it now.
It does go. Yeah, it does come out in various forms.
Right, Claire, over to you.
Good morning, welcome to STEMtastic at West Suffolk College. We're delighted to welcome Dr. Sean Bew, the Senior Lecturer in Chemistry at the UEA, to discuss chemicals are us with us this morning. I'll pass you over. Enjoy the lecture.
Good morning, everybody. I trust you're all well. I'd like to thank, obviously, the invitation to come along today. Hopefully you'll get something out of the kind of session. My plan is to try and kind of give you an overview of what an organic chemist is and why organic chemistry is important. Obviously, with more people living on the planet today than there kind of ever has been, chemistry is more and more coming to the fore with the development of things like new therapeutics, agrochemicals, new materials, new fuels, better batteries. And so organic chemistry is kind of at the core of a lot of this. And what I'd like to do is I'd like to kind of, hopefully you will see my slide change. Is it going to change? My slide isn't changing. Has yours changed?
No, it hasn't. Maybe worth a quick unshare and a reshare if it doesn't.
Yeah, it's not switching over. It was a minute ago, wasn't it?
It was.
Okay, what I'll do is I will come out and then come back in again. Right.
Perfect.
Has it changed? Brilliant. Okay, that's the first hiccup out of the way. Hopefully no more. Okay, so what I was going to talk about for the next hour, but maybe a little bit less, is I'm going to start off with a part one, which is going to be what is a kind of chemist and what do they do and what's an organic chemist, and you know, what, a why, a who, a how, and a time type of scenario. And then I'll kind of give an overview of the different types of jobs you can get. So what I thought would be quite interesting is instead of just talking completely about chemistry, is to maybe give an overview of what job descriptions organic chemists can come under. And it's quite varied. And I've tried to kind of give you some indication of how diverse organic chemists, if you do organic chemistry as a degree, how diverse the job opportunities really can be. And then the second bit, I'm going to talk about kind of real world problems, so a bit more to do with chemistry. So I'll talk a little bit about some chirality and the development of new therapeutics. And I'll talk a little bit more about the development of a new therapeutic which has just gone into clinical trials for COVID. So again, trying to kind of keep it quite topical. And then I'll move over to, instead of being small man-made molecules, I'll move over towards natural product type molecules. And again, we'll talk a little bit about that. Include a little bit on toxic organic chemistry. So again, I won't divulge too much there, but I found out some quite interesting facts when I was researching this and thought I'd kind of include them. And then right at the very end, we might get chance, we might not. I'm just going to put in the 25-year challenge for organic chemistry and what's ahead for the next 25 to 30 years. Okay, so I've kind of got a question for you, and I'm gonna basically ask it now. Which of these is a chemist? This is a kick-off question, and I'm going to put up, hopefully, you should see two slides or two kind of groups of people. On the left-hand side, you've got guys who are working in glove boxes, and there's a guy holding a small vial up in the air, and they're both intently staring at it. And then a bit further down, there's a lady in front of a kind of yellow looking fume cupboard. And on the right hand side, we've got some people with kind of light coats on and lots of shelving and lots of boxes. Looks like there's lots of maybe chemicals on the shelves. And the question is, which of these is a chemist? So is it the guys on the left or is it the guys on the right? And I'm sure you probably all got this, but it's the guys on the left that are the chemist, and the guys on the right are not chemists. So this is a kind of, I guess, a bugbear of chemists, that they get confused with pharmacists. So the guys on the right are pharmacists. They hand out the products that are made by the chemists on the left-hand side. And you can normally tell the chemists because their lab coats are usually slightly ruffled. They're slightly dirtier. They've always got safety specs on, as you'll see here. All of these guys got safety specs on, so safety is very important. And they're usually working in quite cluttered environments. They're not usually very clean environments per se. You know, there's lots of bottles and lots of tubes and lots of big bits of equipment and stuff. And as you can see in the lower right-hand corner there, the lady in front of the yellow fume cupboard, she's using an automated robot with inside the fume cupboard there to handle radioactive compounds. So in a way, completely different to what pharmacists do. Okay. So what does a chemist do? I mean, it sounds like a kind of pretty obvious question, I guess, really. And, you know, as a chemist, it's one that I don't really think about, to be honest, because I do it every day. So it's not something that I really think about. So I had to think about this. And I think first and foremost, chemists study and make new chemical compounds. In fact, this is probably the one thing which drives a lot of, like myself, organic synthetic chemists. It's what drives you to do the job you do. So, you know, we often make new compounds. We make compounds that have never been made before. We test their properties. We look at what they do. And we kind of resynthesize and we tweak them. And there's a genuine excitement when you know that you're the first person to have ever made a new compound. So what's called an NCE, a new chemical entity. It's the only sample on the planet. It's never been made before. And you get all the data and you know that your flask or your vial is the only sample in the planet that's got this compound in. That's quite a unique experience to actually know that you've developed something that's never been made before. So organic compounds are used in, you know, many different areas. So they're used in therapeutics, They're used as dyes in printers. So you want your blue, your yellows and your reds in the printers. Those are based on organic compounds. Petrol in cars is obviously organic. Materials for planes, you need it to be lighter. So again, organic based compounds a lot here. So the question is then, what skills do you need to have as a research chemist? And I've kind of listed some on the left-hand side there. And this is probably not an exhaustive list, but it does give you a kind of idea. And I just want to kind of say a few words about each one, because if you think you've got the kind of attributes which fit these kind of skills, then you probably make a very, very good chemist. Not necessarily, you could be organic, you could be inorganic, you could be analytical, but these are generally the things which a chemist, the kind of attributes that a chemist would have. So patience. If you're doing research, you've got to be patient, especially with what's called blue sky or fundamental research. This is often quite slow, but again, at the end result is usually very rewarding. Flexibility. So this doesn't mean you can touch your toes. This more means that you've got mental flexibility, you're open-minded, you're prepared to accept new ideas. Most chemists are numerate, so they're good or relatively good with handling numbers. They're logical. So not only are they good with their hands, as you saw in the kind of previous slide where they were in the laboratory, but they're also quite good with maths and putting numbers together. And with a degree, a chemistry degree, what you'll find is you generally find that you'll get taught to look at things in a logical sequence of processes and to break things down into smaller bite-sized chunks. So we tend to try and encourage the students that we teach to say, this is the big problem, but the bigger problem is made up of smaller problems. If we dissect the smaller problems, we can try and come to a solution to the bigger problem. So this kind of breakdown into logical bits is a kind of way that we teach. Analytical, obviously being good attention to detail, precise record taking, noting data, observations. These are all very, very good characteristics. And more so in the 21st century is what's called interdisciplinary or soft skills, the interdisciplinary skills, communication skills, IT skills. These are often called soft skills, presentation skills. These are also really important, and more so in terms of the kind of job descriptions that now come up more and more often. So you can see on the right-hand side there, I've got kind of typical employers, again, not an exhaustive list, but again, quite a diverse. And the one or two that are missing off there, for example, is lots of chemists go work in biotechs and medtechs, as well as patent attorneys. Chemists make very good patent attorneys for the reasons I've just alluded to. They're good at logical breaking things down into small bite-sized chunks and putting an argument together, which is also logical. So what flavors of chemist are there? So on the right-hand side, I'm going to start off with the chemical biology on the right-hand side here. So chemical biology is where you use chemistry to probe biological processes. And this has really come to the fore in probably the last 10, 15 years or so. So the development of specific chemical probes which answer a specific biological type question. And underneath there, you can see that I've got a question, which is Lord Percy, and this will become evident when I've asked this in a minute. Is Lord Percy an alchemist or a green chemist? Now, I think you can see that there's a poll on the screen, and it asks you basically the same it asks you the same question: is Lord Percy an alchemist or a chemist? And you can click one other. This is just a bit of fun. There's nothing serious about this, but it's to do with the video. And I'm going to show this video now. It's only a couple of minutes. And then we'll come back. Hopefully this video will work. There's no point making something if you don't know what you've made. And there's also analysis, which is purification. There's no point making something if it's not pure, because the properties won't be what you're actually genuinely seeing. Materials chemistry, you can see I've kind of highlighted this with the kind of broken bones. So here we're using graphene to help mend broken bones. Again, this is materials-based chemistry. And at the top, we've got medicinal chemistry and natural products. So the structure of vancomycin is a natural product, which is isolated from the soils of the jungles in Borneo. And this, again, is really coming to the fore, and we'll see a couple of examples of this in a few moments. So what is organic chemistry? So I'm not sure if you've done much organic chemistry at school at the moment, but the definition is really quite simple, really. It's the chemistry of carbon-containing compounds. So they don't only contain carbon, they often contain sulfur, phosphorus, bits of chlorine, bromine, fluorine, nitrogen, and obviously oxygen is a common one. But what makes carbon and organic chemistry unique is that carbon is basically the only element that can really form branches with itself. So it can form branches, as in linear chains, but it can also form cyclic molecules. So if we just look at the simplest example on the left-hand side there, you can see methane in blue. And methane is obviously the greenhouse gas. It's widely available, if you like, on the planet. It's not the best source for generating compounds because it's actually relatively unreactive. But we could imagine if we could take methane and add 8 carbon-based methanes together, then you can see next to methane, I've drawn out that kind of chain with the CH3, CH2, CH2, repeat, 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 and we get petrol. Okay, so we've gone from a gas, which is relatively abundant, and we've made petrol, and we all know how useful petrol is. If we do the same process, but instead of stopping at 8, we actually go to 10, and we add an extra 2 carbons in, then we get to kerosene, and kerosene is the fuel that's used in aeroplanes. And similarly, if we go from methane again, but now we go to 12, we end up with diesel, again, a fuel. So organic chemistry is really important for developing fuels, for enabling us to traverse the planet like we've never been able to do before. On the flip side, instead of having a linear chain, we could actually have a cyclic chain. So you can see on the left there, we've got cyclohexane ring, which has got 6 carbons in it. You can count them round from the top. And this is a really important compound because it's used to make nylon. So again, now we're talking about making clothes and plastics. And if we take hydrogen out of cyclohexane, then we end up with benzene. So you can see the arrow that moves to the right. And if we generate benzene, benzene is a common, or the ring type structures that benzene is made of are commonly found in drugs. So again, really important compounds. And I've given one final example that if we could take 2 methanes, you can see there the red one and the blue one, but now we're going to add a heteroatom, in this case we're going to add water, then what we get is the molecule on the right, and I've kind of color coded it, and you should be able to follow where the carbons from the methanes have ended up inside our molecule. And you probably know that this is ethanol, and we all enjoy a little drop of ethanol every now and again, I'm sure. But you know, if you think about it even more, then we just simply cannot get away from the fact that organic chemistry is everywhere. It's at the heart of everything we do every single day. So I've kind of given this typical day, if you like, along the bottom here, where we use or we have involvement with organic-based compounds. So we wake up in the morning and we have a shower and we use some soap. And of course, soap is made from the saponification of fats. So I've got an organic-based reaction there to make an organic-based compound. We maybe have a cup of coffee on the way to work and we don't want to use sugar, again, an organic compound, but what we're going to do is we're going to put in an artificial sweetener. So in this case, we're going to use canderel, and canderel is made up of 2 amino acids. It's made up of aspartic acid and phenylalanine, and it's about 200 times sweeter than normal sugar. We get to work and we sit in our chair, and our chair has a plastic back mesh and maybe plastic armchairs, handles, maybe feet, which is obviously derived from the petrochemical industry. So again, carbon-based. We open our laptop, we use our laptop, and the motherboard within our laptop is held together using epoxy resins, keep everything in place. Then we go home, and on the way home, we maybe stop off for some food. Maybe you go to McDonald's or somewhere like that, and you have a burger. Well, the burger you're going to eat, you sometimes have it rare, sometimes you have it well done, and there's a reaction going on the surface of the meat there, which is called a Maillard reaction. And when amino acids and sugars are heated up to high temperatures, they undergo a condensation reaction and you form a new product. And depending on how long you heat that reaction and how hard you heat that reaction, depends how hard the organic reaction proceeds, which defines how brown your meat goes. So the brownness is connected to the organic reaction which is happening on the surface of your food. Maybe you don't have, maybe it's a Friday and you have fish and chips. So the vinegar you put on your fish and chips is acetic acid, a weak organic acid. You're now nicely replete, so you sit in front of your OLED TV, which is organic light emitting diodes. These are lighter, thinner, less energy intensive TVs. And then, of course, whilst you're watching TV, you might have a pint of beer. And of course, beer chemistry, you can do a degree in beer chemistry. There's so much organic chemistry going on in there, they've done a degree on it. And of course, if you have too many beers, then you're going to need a paracetamol, which is an organic based compound to cure your hangover. So every single stage of our day, we're generally in contact with some organic based compound. So I wanted to kind of talk to you about, I started to look into this, and I know this guy's got a guy called Phil Barron, and he's at hopefully this will work. Hopefully you can see this, but I'll let Phil talk to you about what he thinks an organic chemist is.
My name is Phil Barron. I'm in the Department of Chemistry at the Scripps Research Institute. I'm an organic chemist and an educator. And right now, our research is focused on simplifying the way molecules are made for the purposes of pharmaceutical industry and material science and agrochemicals. I have always been obsessed with chemistry. The area of organic chemistry is such a beautiful one because one can be both an artist and an explorer at the same time. So you can paint and you can visit the moon, but you don't have to leave your laboratory to do that. So the beauty and science of the field of total synthesis is one that manifests itself in the unpredictabilities and new things that one discovers because we just don't understand fully the language of molecules. There are a couple of recent examples that excite us. So one is this one. This is a sort of plastic model depiction of a molecule known as cortistatin A. It's a molecule that was isolated from a marine sponge, and it was found to have miraculous bioactivity. And it became a sort of worldwide hunt to try to recreate this molecule in the laboratory, because the only other way to harvest it would be to destroy an entire ecosystem on the bottom of the ocean. And even that wouldn't be a sustainable solution. So our laboratory a few years ago devised a way of making this compound in a practical fashion, because in general, the bottleneck is not demonstrating the feasibility of constructing a molecule like this. The bottleneck to applicability is being able to hold a fistful of it in your hand. You know, with natural products, it's kind of like a tease. Mother Nature shows the structure of a compound and delivers it, and you can tell that it might be useful for something. In this particular case, because it is marine derived, very little of it can be made. So if you wanted to turn a molecule like this into a potential anti-cancer treatment, the only way you could actually do that would be to make a lot of it. So by producing it, you learn something fundamentally about organic chemistry, but at the same time, you make it into a real, tangible product that can actually be evaluated as a potential medicine. What's exciting about this, to me, is the application to society. What the foundation support will allow us to do is pursue ideas which probably wouldn't be fundable without already showing that it's going to work. So it will just allow us to accelerate the process of discovery. In that regard, for us, it's just an incredible opportunity.
Okay, so that was Phil's take on what an organic chemist is, and I think he summed it up quite nicely there with the fact that we all make compounds which have generally are useful in across the board, whether it be drug synthesis or agrochemicals or plastics or anything. Organic chemists have generally got an input into it. So what training do you need to be an organic chemist? So what I've done is it's kind of slightly complicated, but hopefully I can kind of suggest how actually easy this is to understand. So on the left-hand side, we've got the typical salaries, and these are just ballpark figures. These are just, I looked at GSK's, GlaxoSmithKline's website, and I kind of pulled these off their website. So, you know, they're not set in stone, and certainly not when you move further down the screen to where you have we become a doctor. The salaries do kind of, there is no gap really. You can there's no cap on what your salary will be once you become a doctor, but it gives you a vague idea of the kind of ballpark figures. And in the middle, we've got the kind of route that a classic chemist would take. And on the right-hand side, we've kind of got the job descriptions and what you would end up generally doing if you was to drop out from degree. You would go into work, or you do a PhD, and then you go into work. Those are the kind of levels. So I'll go through it from top to bottom. So the classic scientist A-levels, I guess, is chemistry, physics, biology, or chemistry, physics, maths, or chem biomaths. These are the types of degrees, A-levels, that would get you into a chemistry degree program. You can generally do BScs or MChems, and the real difference between the two is that the BSc is 3 years and an MChem is 4. Usually the MChem is the 4th year is more advanced chemistry, and you can usually fine tune what you want to do. So maybe in the 4th year, you'd prefer to do some organic chemistry, so you can bias it, your teaching, your research project towards organic chemistry. And also sometimes what you can do is you can do what's called a year in industry. So you go away from the university for the best part of a year, and you work and you get industrial experience, you know, in a big pharmaceutical company or an agrochemical company or an analysis company. And the benefit of this is that you get paid. So you get paid, you know, pretty well generally. So you're still a student. You have all the kind of benefits of being a student, but you get paid maybe 17, 18, 19,000 pounds a year. So that's not bad for being a student. That's pretty good. If you leave after you do your chem and you go work in industry, say GlaxoSmithKline as your company, you'll predominantly stay at the bench. You'll be what's called a bench chemist, and your job will be to make new compounds. You probably won't do too much managerial type work. You'll predominantly just stay in the laboratory. Some people can't decide whether they want to do PhDs or not, so they do an MSc, which is a 1-year research project, which gives you a flavour of what it's like to do a proper research project for a whole year. And again, if you decide PhD is not for you and you leave, then you'd go to work industry, and you'd probably be be the bench chemist. You'd spend most of your time at the bench. If, however, you go and decide you want to do a PhD, then so PhDs take 3.5 to 4.5 years. These are almost exclusively research-based only, so you'll be based in a lab doing a research project. You are funded externally, so you don't pay for it yourself. You probably could pay for it yourself, but generally PhDs are funded externally, either by government or by companies. The salaries for PhD students are around 15,000, a little bit more, but there are exceptions. So, for example, the Wellcome Trust gives up to, I think it's up to 23,000 or 25,000 pounds per year. So again, that's not a bad salary, and you're still a student, so you don't pay tax. At the end of your PhD, you write a thesis or a big book, which is probably going to be 200 or 300 pages long. And then you have what's called a viva, which is where you sit basically in a room with you and 2 examiners, and they talk about your project. And this can last for 2, 3, 4 hours. And then there's basically a pass or fail based on your book and how you did in your viva. After you do your PhD, you can leave again, you go to work in industry, but this time you'd probably go into do some laboratory work to start with, but then the emphasis is usually on moving you towards managerial type, senior scientist level or team leader. So you'd probably have people working for you. And then the alternative one is that you stay and you do what's called a PDRA. So PDRA is a postdoctoral research associate, which again, these can vary. You get career PDRAs who basically they just stay as postdoctoral researchers for the whole of their career inside universities. But most people stay for 2 to 4 years. And once you leave, again, if you go to industry, you're usually fast-tracked to a senior scientist, team leader, or department head. Again, more managerial, kind of looking after people, looking after students and people that work under your wing inside the laboratory. So you can see that to be kind of a fully fledged, if you like, organic chemist with a doctorate and PDRA, if you want to go working in an industrial organic chemistry laboratory environment, that'll be 9 to 10 years. But alternatively, if you want to be working in an academic organic laboratory or an institute, it's slightly longer at 9 to 13.5 years. So a reasonable amount of time, so you can see that it's actually, it's quite an intensive process. Obviously, sometimes if you want to go work in an academic lab, you generally have to kind of work at the big universities, so the Oxfords, the Cambridges. Sometimes you have to go to the US to do postdoctoral work in big research group in the US. But in academic jobs is very, very competitive. So you have to be very good at being able to do organic chemistry. So what I'm going to move on to now is I'm going to that's the kind of part A, if you like, and I'm going to move on to the part B. And I thought I'd just kind of put into context. So by the end of the talk, hopefully you'll kind of realize where we were approximately 100 years ago. So you can see on here, we've got salvarsan, which is an arsenic-based drug, which was developed around the early 1900s, 1910 by some German chemists. And obviously they realized that arsenic was poisonous, was toxic. And so what they tried to do is they tried to wrap organic molecules around the arsenic to try and tame it, to become less toxic and to maybe make it more specific for the diseases of the day, which tended to be things like sepsis, so which is a blood disorder. Now these compounds were very effective, but they were kind of a bit too toxic, to be honest. They would be way too toxic for today's environment. And so, a few years later, you may recognize the structure in the middle there. This is penicillin, in the late 1920s. And of course, penicillin is still around today. So it's obviously very, very useful. It's still very widely used. There was a few problems with penicillin when it was first isolated, and that was purifying it. Again, we come back to the kind of slides we had before. If you can't purify something, you can't really do what you need to do with it. So purification for penicillin took a while to overcome. And then in the 1930s, 1940s, there was a lot of work being done on dyeing cloth, on wool. And again, this compound you see on the right is called an azo dye. And this is a bright red prontosil. And prontosil dyes wool. And so the guys that were doing this kind of made the kind of mental leap that, well, if it dyes wool, and wool is made of amino acids, and bacteria are made of amino acids and proteins and sugars, perhaps the azo dyes would have an effect on bacteria. And this proved to be exactly the case, that when you administered the dye to people that had bacterial infections, the dye killed the bacteria but did not kill the people. It was a really, really selectively toxic process, specifically for bacteria. So this was a big, big leap in terms of organic chemistry and the targeting and making of organic molecules into medicinal compounds, which had selective biological activity. So the question is, how do we start to develop a new drug? That's where we were, 60, 70, 80, 90 years ago. What does it look like now? And basically, it's very, very difficult. To make a new drug from scratch is a very, very difficult process. So you can see on the left there, I kind of put in again, do you remember what I said about the kind of teamwork and the soft skills? You've got to be able to work with people who don't speak your language as a chemist. So structural biologists, molecular biologists, pharmacologists. If you want to be a medicinal chemist, you've got to be able to understand what these guys are talking about when they tell you that their protein isn't doing what you think you would expect it to do. So again, you've got to understand, this is one of the benefits of chemists, is that chemists can generally understand biology. This is one of the benefits of actually being a chemist. And on the right hand side there, you can see I've got this kind of roadmap of how drugs are developed. And you can see it's like snakes and ladders. You kind of progress up the road, and then there's a problem and you kind of fall back down to where you were before, and that's probably where the drug has failed. It's not met what it was supposed to do at that particular point, so you need to resynthesize it or remake it and change it. And you can see that effectively we kind of go through various phases. You have phase 1, phase 2, which is where it starts to go into people, and then it goes phase 3, whereas it goes into lots of people all over the planet, which is drug testing. And then finally all this data is analyzed, and it's kind of given a thumbs up, if you like, by the FDA or the EMA, the European Medicines Agency. So this is a very, very long process, and it's also hugely expensive. So you can see the average time on the left there is 10 years to develop a new drug. That is a very, very long time for any new drug to be developed, especially if people are dying, you know, in increasing numbers. And the cost to developing new drugs is enormous. So easily runs into billions of pounds, and that's per drug. So it's a fantastically expensive process. So the question is why does it take 10 years to develop a drug? And we can kind of go back, if we trace back to the 1950s, there was a new wonder drug, as it was known at the time, called thalidomide, and you probably will have heard of thalidomide. And thalidomide was given to pregnant women to try and stop nausea. And of course, they didn't realize at the time that when the babies were born, they had shortened arms and legs, and this was directly attributable to the drug that they were given, the thalidomide drug. So you can see the structure of the compound in the middle there. That's thalidomide, the compound, and we'll talk a little bit about that in a minute. But we need to kind of understand why it caused the problems it did. And this is based on the fact that thalidomide was a chiral compound. And so I'm not sure if you've heard of chirality. You may have done, but if not, then I'll just quite quickly give you an overview of what chirality is. If you imagine putting your hands out in front of you and you have it so your both hands are either palm up or palm down, as long as they're the same way, it doesn't matter which way you do it. If you have them both be the palms up and you put them both in front of you, you'll notice that your thumb on your left hand points to the left and the thumb on your right points to the right. If you now try and pass them over the top of each other, so you place them over the top of each other, but you keep the palms pointing up, you'll notice now that you cannot get your thumbs so that they are both in the same position and the fingers and the thumbs are all locked. You can't do it. You can't change it. You have to flick your hand over, and if you flick it over, then you can. But now it's a mirror image of the other finger, the other hand, sorry. And this is the property of chirality. It's a mirror plane. And so chemicals have a mirror plane. They have a handedness. They have a left-handedness and they have a right-handedness. And in fact, the problem with the thalidomide was that it was administered as a 50% mixture of the left hand and a 50% mixture of the right hand. It wasn't given as a single left-handed or a single right-handed compound. And one of the compounds, one of the hands, was actually toxic. And this is what caused the problems. And it was recently found that the 2009 that the problem with thalidomide was that it cut down the blood supply to the growing arms and legs. So as the arms and legs of the baby were growing, with the drug stopped the blood supply going to the arms and the legs, which is why, of course, they didn't grow properly. So here we can see that there's I've just tried to kind of again show you the kind of the property of chirality. In this case now, I've shown it in the context of the thalidomide, and you can see that the structure on the left of the kind of mirror plane in the middle is what's known as the R thalidomide, and this was the correct one. This is the one that induced the sleep and also helped with nausea. And you can see I've labelled that R, and then you can see a wedge and a dash, and the carbon which that is attached to is the chiral center. And in this example, that carbon there has R configuration. And on the flip side, on the mirror plane, you can see that the S now is the enantiomer, as these are known, the hand. This is now the one which is tetragenic. This is the one that causes the problems. So if they'd given thalidomide as only one hand, and the R hand was only given, they had tested the compound, the left hand and the right hand separately, they'd have worked out that actually thalidomide would have been a problem, and then they could have got around that problem by just giving one of the hands, in this case the R isomer on the left hand side, and we would never have had the problem. Now this because of this issue, the pharmaceutical industry is very, very cautious when it has chiral compounds and administering them as drugs to the general public, and it needs to know that its compounds are absolutely 100% safe. And because this takes so long, this impacts the amount of time it takes to get the drugs out, and hence why on average most drugs take about 10 years to get to market. So a little bit more about chirality. Chirality is everywhere. We're surrounded by chirality. If you imagine you're sat in a left-hand drive car, it is chiral compared to a right-hand drive car. You cannot switch a left-hand drive car to a right-hand drive car without effectively breaking the steering wheel off and sticking it on the other side. So you've broken the car down. And the same way as with organic compounds, if you want to convert a left-handed molecule to a right-handed molecule, the only way you can do that is by cleaving a bond and then reattaching. Scissors on the right-hand side, if a left-handed person tries to cut using right-handed scissors, they find it quite tricky. So scissors are actually chiral. Screws, just screws, simple screws, will only go into wood one way because they've got a chirality to them. The screw thread is chiral. Light bulbs, if you have these kind of bulbs here with the twisted bayonet, they'll only go in one way. So a light bulb is chiral. Golf clubs, left-handed and right-handed golf clubs. And of course, if you try and put your left foot in a right shoe and the right foot in the left, your right foot into a left-handed shoe, you will feel the difference. Similarly, usually with gloves as well. So chirality is a really, really important process and property, which is in organic chemistry is one of the main areas of research that people work in, principally because it's used a lot in the pharmaceutical industry. So different hands, if you like, the left hand and the right hand, have different biological properties, and that's what we have to be wary of when we're making drugs. So it's clearly going to be very important to make a single enantiomer, a single hand of a chiral compound to test to see if it's got any adverse properties, or in fact, sometimes single enantiomers have better properties than when you have the mixture of the 50-50 mixture. You can see that 80% of the FDA, so this is the Drug Administration Agency in the US, 80% of the drugs that are approved are chiral, and 75% are only left or only right. So again, 3/4 of all drugs are left-handed or right-handed. And this is a massive, massive business area. So you can see this is worth $18 billion a year. So it's hugely important. So let's have a look at some examples of some simple drugs. We'll start off with ibuprofen. You've probably heard of ibuprofen, probably even taken ibuprofen. It's a simple pain relief, reduces swelling. But you can see that I've indicated with the hand over the top there, and then you can just see that the kind of wedge that is the S chiral center of ibuprofen. Salbutamol developed 5 years later. Again, now we're talking about an R-handed molecule, so the thumb is now on the opposite side. Salbutamol, if any of you have got asthma, you have a little blue inhaler. This is the chemical that's inside the blue inhaler that you take. Cetirizine, if any of you have got allergies, maybe to pollen, in a couple of months' time, you'll probably be taking cetirizine. This is for antihistamines. Citalopram is used for depression, and you can see now we're getting slightly more complicated molecules. So if you look at compared to maybe the ibuprofen, the chiral center here has no hydrogen attached. It's got 3 carbons and 1 oxygen. This is a quaternary chiral center, and these are quite tricky to make. Again, so you can see this has got an S hand, developed in 2001. Lansoprazole, this is quite a special chiral center because the chirality now is based on sulfur, not on carbon. But again, it's now the S-handed. That was developed not so long ago in 2009. And very recently, you may have heard of this compound, remdesivir, which is the compound that's just been FDA approved for treating COVID. And now you can see that we've got 1, 2, 3, 4, 5, we've got 6 chiral centers on this molecule. So you can see that as we've gone through the years, the chemistry and the molecules we're making, the numbers of chiral centers have become increasingly complicated and increasing numbers of chiral centers. So the question is, do more chiral centers cause more problems? And of course, as with everything in these kind of situations, the answer is yes. So we use a very, very simple formula, which is 2 to the n, where n is the number of stereocenters you have in your molecule. So the number of hands, left or right-handed. So you can see that naproxen at the top there, we've just drawn it out, it's got that on the left-hand side is the R and on the right-hand side is the S, and it's only got 1 single stereochemical center. But if you look at the example below, this has got 2 chiral centers. So if you look at the far left of the 1, 2, 3, 4 structures, you'll see that I've labelled one on R and one of the other ones is S, and then I've drawn the hands above. So now, because it's got 2 chiral centres, we have 2 to the N being 2, means we have 4 different combinations of our hands. We can either have RR, SS, RS, or SR. And each of these is a unique compound. They are not the same molecules. They have unique physical properties, and almost certainly they'll have unique biological properties. So you can see that the introduction of new chiral centers inherently gives us a headache of introducing more and more compounds to have to analyze if you were talking about making a drug. And so, of course, if you have 4 stereocenters in a molecule, and you use our same formula, 2 to the 4, it now means you've got 16 compounds you're having to make. Now we're starting to get a little bit tricky with making, it's going to be a long time, a lot of process, a lot of money to try and make all the 16 compounds, to test them all. Now it's starting to get a little bit tricky. So let's say, for example, that out of our 4 compounds we've got here, that we've decided that the active drug is now, we've analysed and we've determined that the active drug is the RS, and I've highlighted that with this kind of pale blue box. Now, it might be the best drug in the world, but every drug that you take has to be metabolized by your body. And quite often, the problems, and this was part of the problem with the thalidomide, is that you form the toxic metabolites. So the drug itself does exactly what you want it to do. It sits inside an active site and it stops a biological process. But your body needs to get rid of that drug. And so it starts to break it down using the liver. And what normally the body will do is it will add oxygen to the molecule to make it more water soluble, because then it can get soluble in your bodily fluids, which means it can either be excreted or urinated out of the body. And so you can see here what I've done. This is a hypothetical molecule. It doesn't really exist. You can see that the toxic metabolite that's been generated, I've shown with the 6-membered ring has now got this is called a quinone with the carbon oxygen double bond and the 2 double bonds either side. And then next to the aromatic ring, I've inserted an extra OH group. And again, this is the way that the body generates it generates analogues to try and get rid of the molecule that it now wants to get rid of. But this has turned out to be toxic, but we still want to use it as a drug. So we need to find a way of stopping this toxic metabolite formation. And what the medicinal chemist will do is they'll look at the metabolites that have been formed, they'll test them to see if they're toxic, and then if they are, they'll try and stop that toxic metabolite being formed. And these are 2 classic examples of the way this is done. One of them is the incorporation of fluorine. Because the carbon-fluorine bond is so strong, most enzymes that are involved in the metabolism of drugs can't break the carbon-fluorine bond. So by inserting a fluorine bond there, you stop the metabolism at that particular point. And of course, if you just remove the hydrogens that were causing the problem to start with, in this case, I've inserted 2 methyl groups, then of course now we also stop metabolism at that point. So now we have to retest this compound. Is this got the same biological activity as the compound that we originally made at the very beginning? And you can see how this process, this iterative process, makes drug discovery really, really, really tricky, but also quite a long-winded process. So the key to developing new drugs is you've actually got to know what you're trying to target. So if you want to try and, for example, I've shown on the left-hand side here, this is COVID attaching to the spike protein on side a cell. If you want to try and target the COVID, there's no point looking at drugs that interact, for example, with dopamine inside the brain. You've got to know exactly what the structure is of the compound, of the active site within the biological compound you're looking at. So I'm just going to go quickly through this, and this is for COVID, and basically every biological process is not just one process, it's usually like a cascade of processes, like 4, 5, or 6, that all tie up together. And if you can selectively target one of these processes, or maybe 2 of these processes, then you can stop the biological effect, whether it's cancer, or whether it's diabetes, or whether it's obesity, or just something as simple as headaches. So if you can target specific biological parts of this pathway, then you can start to, if you like, unpick the way that the process works. By inserting a drug, you can stop that particular process. So for example, we could try and stop the fusion of the SARS protein onto the surface of the cell using the spike protein. Can we stop that process? That would stop the SARS virus getting inside the cell. That would seem a viable way. Alternatively, we can stop translation. So translation is where the virus RNA is converted into proteins within the cell, and the protein is made as a very long strand, and it needs to be chopped up into smaller parts. And these smaller parts are then reassembled to make more viruses. But the protease, which chops the long protein up, is an enzyme. And so perhaps we can target that protease with a chemical to stop the long protein from being chopped up, which would then, secondary effect of that would be that the virus wouldn't be able to replicate. Alternatively, we can use reverse transcriptase inhibitors, which work on the viral RNA, which is where remdesivir does work. And then finally, you could work on maybe working on the packaging or stopping the virus from being released. So each one of these points, if you can insert a chemical which stops that process, the chances are you can help combat whatever disease you're looking at. So again, as I just mentioned, this was the way that the remdesivir targets the reverse transcriptase, and it stops the RNA from making more RNA, which then goes into packaging, which then is then expelled from the cell to make more viruses. So the process of making drugs, I'm just going to quickly kind of go through this. This is a if you like a kind of 6, maybe a 7-stage process. The first part is we need to know what we're trying to target. So we need to know our target structure. And we're going to look at this in the context of the proteolysis. So remember, the protein that's made by the COVID, that's a long protein which needs to be chopped up into smaller proteins, which then can be transformed into RNA, which then makes more viruses. So if we can target that enzyme, the proteolysis enzyme, we stop that process from happening. So this is the actual structure of the enzyme that we're going to try, of the protein we're going to try and target. And you can see that I put on a kind of purple star. That's the active site. That's the point where all the activity in terms of proteolysis occurs onside the protein. Now this protein weighs about 60,000 kilodaltons, so it's reasonably big. That's a pretty big proton. When you think that most drugs probably weigh about 500, 60,000 is a big difference. And your drug has got to scan that surface of the protein, it's got to find the active sites, and then it's got to get into the active site, and it's got to fit into the active site like a hand in a glove. Because once it sits inside the active site, it's going to stop the natural process which would normally happen from occurring. And so you can see that I've kind of expanded on the left-hand side there. There's a kind of rectangle, and I've put the star at the point of the active site on the surface of the protein. And you'll see the proteins are slightly different colored. Some are blue, some are brown and gray and red. And this is to do with the type of amino acid at that point. So whether the amino acid is acidic or basic, this is going to impact how your drug will interact. So they used the most powerful computer in Germany for the drug discovery. The escalates its 4COV program, and this molecule was scanning. Sorry, this computer was scanning 5 million compounds every second, and they tested 70 billion compounds. So what they do is they take the protein structure and they computationally see if they can match the chemicals with the protein to see if they can find ones that will sit inside the active site. And they tested 70 billion compounds. Out of those 70 billion compounds, they got a relatively small number which seemed to fit inside the active site. So they get handed on to the organic chemist, and the organic chemist does what's called high throughput screening. So you can see we've got some robots here, and the robots make maybe several thousand analogs of the ones that have been shown to be in the computer to potentially sit inside the active site. These then are then co-crystallized with the protein. So you take each one of those compounds and you add it to a small amount of the protein and you try and get a crystal structure. And the crystal structure will tell you exactly where the compound is sitting, and it will tell you exactly where it's inside the active site or not, and how good a fit it is inside the active site. So again, is it a really snug fit, which is what you actually want? Good interactions, nice and strong. So you can see on the right-hand side there, we've got the kind of grey kind of cloud, and then inside there, we've got the molecule, which is now sat inside our active site, and it's a pretty good fit. It's not the best fit, but it's okay, it's not bad. So what we need to do now is we now to bring the organic chemists back in. They're now going to resynthesize the molecule, and they're going to look at the crystal structure, and they're going to try and make it fit even better, have more and more stronger and stronger interactions. And then finally, what we do now is we want to test this compound to see if it's got any what's called ADME properties, so absorption, distribution, metabolism, excretion, and toxicity problems. And again, any issues that arrive here with the compound being toxic or not being water soluble enough, they'll go back to the chemist will resynthesize the compounds, and then they'll get what's called biologically evaluated. And these now are known as hits or lead compounds. And they will go into usually rats or mice, sometimes dogs, pigs, or monkeys for proper evaluations of the toxicity issues. And if they come through those studies, then you have what's called a drug candidate. And this molecule here is a genuine drug candidate that was developed using this exact process, developed by Pfizer. And if you remember, we said that most drugs take 10 years to make. Pfizer made this in about 7 or 8 months. So they absolutely flew through this process and came up with a compound which is now in clinical trials. So this has gone to, it's in man. It went into man in September. They put it into 70 odd patients, and the results will be known in July. If they're good, then it goes to what's called phase 2, which is where it will be tested under more rigorous conditions, and if it passes phase 2, it then goes into phase 3. Phase 3 trials are global. There will be thousands of people all over the globe that will be taking this compound, and they'll be kept under close supervision to see if there's any adverse reactions. And if it passes that, then it goes to the FDA, and then if the FDA like the data, they will give it the thumbs up. So this is a very, very long process, and I think hopefully you can see now why it takes so long normally to do this kind of chemistry. So that was kind of developing chemicals from, if you like, from the bottom up, from simple starter materials. Of course, nature is a source of inspiration too, and I've kind of given a few examples on here. So for example, reserpine was isolated from the Indian snake roots in 1952, and this is used as for high blood pressure. You've all heard of cocaine, of course, but it's actually a very good painkiller. And also morphine, which comes from opium, again, is an analgesic. Taxol, this compound on the just at the top right there, this is a very strong anti-cancer compound, which comes from the bark of a yew tree. And also the ocean is a very, very rich source of what are called metabolites. You can see some examples that I've kind of put up here. The discodermalide is an anti-cancer compound, and the red tide in the middle of that's the Gulf red tide. It produces this compound hemibrevetoxin, which is quite an impressive looking molecule. But one of the most toxic, one of the most complex compounds ever isolated is this compound here, metotoxin. And this is an absolutely phenomenally complicated molecule. It's got 32 rings and 98 stereocenters. And they isolated 20 milligrams. So that is probably like 10 grains of sugar. It's absolutely tiny amount of compound. But they isolate that 20 milligrams from 4 tons of culture. So, you know, this is there in a tiny, tiny amount. The interesting question, though, is how many stereoisomers would this compound have? It's got 98 stereocenters. If we use our formula, and we have 2 to the 2 and 2 to the 4 gave us 16, if we have 2 to the 98 stereocenters, we have 30 with 29 zeros after it, number of stereocenters. Just an unbelievably large number. which is, it's phenomenally complicated molecule to make. But there are groups around the world which are trying to make this compound right at this very moment. The other thing which is interesting about these compounds is they're usually quite toxic, obviously because they have biological effect. And so I'm just going to kind of give you an overview now of kind of toxicity and a comparison maybe to give you a feel for how toxic some compounds can be. So at the top here we have sodium cyanide. So if we think of the average human as weighing 75 kilograms, then one paracetamol tablet would kill the average human. So that's about 450 mg, 500 milligrams. So that's not, if you small tablet, that's pretty toxic. Tetrodotoxin, which is the compound which is found inside pufferfish. This is a Japanese, pufferfish is a Japanese food. It's only 22 milligrams, so a 20th of a paracetamol tablet. So take a paracetamol tablet and divide it up 20 times, and then you'll get 22 milligrams. That would kill you. If you took that compound on the right there, that would be enough to kill the average human. Now look at mycotoxin, 75 kilogram human again, but now 0.0097 of a milligram. So a fantastically small amount of compound. 22 milligrams is kind of tangible. I can picture what 22 milligrams would look like. 0.0097 of a milligram, I couldn't even contemplate how small that is. And that's a 46,000th. So taking a paracetamol tablet and breaking it up into 46,000 bits. That amount of powder, whatever that would be enough to kill you of mycotoxin. So it's an unbelievably toxic compound. But it's got nothing on botulinum. And here you can see you divide your paracetamol tablet 6 million times. So take your paracetamol and divide it up 6 million times, and that would still be enough to kill you. And in fact, if you made 1 kilogram, which is if you make a bag of sugar weighs 1 kilogram, if you had 1 kilogram of botulinum, that would be enough to kill everybody on the planet. So it is a fantastically toxic compound, the deadliest compound known to man. Either way, mycotoxin or botulism, I wouldn't really want to handle these compounds because they would be pretty nasty compounds to go wrong with. So what's the next challenge for organic chemistry for the next 25 years? What we're looking at, and the biggest thing that's going to come after COVID is antimicrobial resistance. So you can see on the right hand side there, I've got that kind of spider diagram, and at the moment there's about 700,000 people dying from antimicrobial resistance, but by 2050, so 20-30 years, that will go up to 10 million people. It will outstrip cancer in terms of the number of people that are dying. And the problem is that people will die from really, really simple problems. So they could go to hospital for a cataracts operation and they could die. And as Matt Hancock said here, people could get paper cuts and they could die. This is a really serious problem. So the problem has been is that antibiotics were made in lots of examples of antibiotics were made up till about the 1980s. You can see at the bottom there, it's been punctuated in the timeline of the last nearly 100 years or so when antibiotics were made, lots of them. And some of these are still available and some of them are still really effective. But the bacteria have now become resistant to the majority of the antibiotics we have. And so this is a serious problem. This slide here gives you kind of an overview of the structures, and you'll notice that I've kind of highlighted one of the compounds is this glycopeptide, and this structure here is one you've seen a couple of times. This is vancomycin, and this was isolated from the soil in the jungles of Borneo. So again, it shows you how nature influences how drugs can kind of start off in a soil, and then suddenly we can be using them as antibiotics. In a kind of literally across the kind of prescription desk. And vancomycin is often referred to as the antibiotic of last resort. So the chances are that if you've got a disease, you've got bacterial resistance, and you've got bacterial resistance to vancomycin, then your outlook is very, very poor. The chances are that you will die if you don't respond to vancomycin. And so with that in mind, people are trying to make new antibiotics based on the ones that they already know are actually pretty effective. So here you can see there's a kind of a graph that basically gives you an idea of the problem we're facing. Is that in 1989 to 2001, the percentage of people that were becoming the resistance to vancomycin inside intensive care, which is where people are at their most vulnerable, really started to rise very dramatically. So this is the molecule down the bottom here, vancomycin, and in the last couple of years, there's a scientist called Dale Boga over in the US, and he started to look at making analogues of vancomycin because it's already got a proven track record. And you can see that he's made one which is 100 times more active just by playing around with the structure. And this is again, he's an organic chemist. He started to play with the molecule, look at the chemistry that it does and the biology that it does, and try to fit new bits onto the molecule which will change its properties to allow it to become more potent. And then last year, he published a paper showing that he'd made a compound which was 25,000 times more active. Again, this was a real achievement, and has spurred on a lot of people now to try and look at vancomycin as potentially having a second life, if you like, of overcoming antimicrobial resistance. And in fact, my group are looking at doing this exact process. So you can see at the top structure there inside that square, most people have been looking either at, like Dale was looking at playing with the sugars, or what the amino acids, which are further down on the vancomycin. And we're looking, as I've highlighted, we're looking at the COD and the DOE rings to see if we can play with these groups and enhance its antimicrobial resistance.
Sorry to interrupt. I'm going to have to wrap the session up as we need to move on to the next one. But I just wanted to say thank you very much to Dr. Sean Bew for his fantastic presentation, and I'll let him finish up. Thank you.
And last slide, that was it.
Brilliant timing on my part there. Thank you so much for giving us the lecture today. It's been incredibly interesting, and my students say thank you from here at West Suffolk. We look forward to seeing the rest of STEMtastic. Thank you very much.
Thanks a lot. See you guys.